Welcome to my channel, The BioBridge. Please hit like and subscribe button for more content like this and leave a suggestion to support me.
Hello, good morning everybody. In last class, we have already studied about anther, parts of flower and microsporogenesis. Hope you have done your revision and is ready for the next class. Today we are going to study about ovule, megasporangia and megasporogenesis. So let's get started with this. First we are going to study about ovule and its different parts. So what is ovule? Ovule is basically the female reproductive part which encloses the female gamete that is present in ovary, and female gamete is female gamete is known as egg. So let's get started. In ovule, in ovule we generally study about the structure that has two ends, that is micropylar end and the another is chalazal end. This is the structure of ovule, right? This is your chalazal end. This is your chalazal end. And this is your micropylar end. What is the significance of these both ends? Micropylar end is that part from where the pollen tube enters into the ovary. It basically guides the pollen tube towards the ovule so that it reaches the ovary and fertilizes the egg. And chalazal end is the opposite to that of micropylar end. So this is micropylar, this is chalazal. Near to micropylar, you can find hilum. It is that part which connects the ovule with the funicle. Funicle is stalk, swollen stalk, which connects this part to the flower. After this, we have these two foldings that is known as integuments. Integuments basically surrounds the ovule. So to protect them, it envelops the ovule. Here we have ovary. Ovary encloses egg, and it has another total 8 type of, and it has total 7 types of cell which we'll deal with later. And one of the most important part that we are going to study here is parenchymatous cell that is enclosed inside here. These parenchymatous cell is basically for nutrition. In megasporogenesis or in megaspore, you have learned that the nutrition provided for pollen tube or for pollen grain formation is taped up. But here, this nutritive layer of parenchymatous cell is nucellus. Nucellus is nutritive layer, right? Generally, this nucellus degenerates at the time of seed formation. Till the seed is formed, the nucellus will not be there, it will be degenerated. But if nucellus is still persistent in the seed formation, then it is known as perisperm. What it is said? Perisperm, that is persistent nucellus. Okay, this is persistent nucellus. Based on the number of integuments present or the structure that integuments is possessing, it is said to be have of 5 types. First is unitegument, where the integument is said to be in single layer. A single envelope surrounds the ovule. So a single integument surrounds the ovary. Example of this is synpetalous and gamopetalous dicot. Another type is bitegmic. Here, integument, like you are seeing here, integument is present in 2 envelopes, in 2 layers around the ovary. So, wherever the 2 layers of integument is present, it is said to be bitegmic. Example is monocots and polypetalous dicot. Okay. Another one is aril. Aril is collar-like integument. Integument present as a collar. Example of this is lychee, nutmeg. Next is carnacle. Here, integument forms the another outgrowth around the micropylar end. Example of this is castor and Euphorbiaceae family member. And last is ategmic, where there is no integument present. Integument here is absent. So example of this is Loranthus, etc. So the structure of ovule that you are seeing here is anatropous. Now you will ask, what is anatropous? So on the basis of structure, how this micropylar and chalazal end is arranged, there are 6 types of ovule structures that we are going to discuss. This type, you remember, it is most common. This is anatropous. Okay. Now we are going to study about types of ovule. Types of ovule. There are basically 6 types. First is orthotropous. Orthotropous, where chalazal end and micropylar end lies in single plane. They are lying in single plane. This is your chalazal end. Chalazal end is CE, and this is your micropylar end. They are lying in single plane, straight line. So this is your M. Okay. So in orthotropous, chalazal and micropylar end lies in single plane. Another name for orthotropous is atropous. An example for this is Piper polygonum. Okay. Another type is anatropous that you have seen right now. Anatropous. What happens here is this particular structure takes a 180-degree turn. Means this particular chalazal end comes towards the micropylar end, and micropylar end comes towards the chalazal end. So it looks like an overturned structure. This structure that you have seen is basically this is your chalazal end and this is your micropylar end. So this is the anatropous type, right? It is most common. So any flower or any angiosperm that you see around you is an example of anatropous ovule. Third type is hemianatropous. Here 180-degree turn is there. Hemi is half. So here there will be 90-degree turn only. Okay, here there will be 90-degree turn. So it will look like this. This is your micropylar. This is chalazal. Micropylar and chalazal are at 90-degree angle to each other. Example of this is ranunculus. Ranunculus you have studied in 11th class. The common name of ranunculus is buttercup. And where you have studied this name? Are you able to recall it? Heterophyly. It shows different type of structure, physical structure in aquatic form and in terrestrial form, right? Remember, in plant growth and development chapter. Okay. Next, moving on to the fourth one, there is Compylotropous. In hemitropous, you are able to see that chalazal and micropylar end is at right angle to each other. But in Compylotropous, what happens is it is either less than 90 degree or more than 90 degree, not at exactly 90-degree angle. Okay, then it is said to be compilotropous. Example of this is Cruciferae family. Okay, next is Amphitropous, where the ovule makes its shape like horseshoe. You will say horseshoe-like structure here of ovule. This is known as Amphitropous ovule. Example of this is poppy flower. Poppy is Papaver somnifera that is extensively used as opoids. And last one, not the least, is Sarkinotropous. Now listen to me carefully. Sarkinotropous. What happens here is first the ovule is in orthotropous position, right? This is your orthotropous. Then it takes a turn and forms anatropous. Now 180-degree turn is taken, so then it is forming anatropous, right? 180-degree turn. And after this, it again forms orthotropous. So it will take a turn of 180 degree again. So there is a layer of cunicle around it, surrounded, wrapped. So it is known as sarkinotropous. Example of this is opuntia. So these are the different structures. So these are the different structures of ovule based on their position and how they are arranged itself. First one is orthotropous. Orthotropous is in single line. Example is polygonum or piper. Anatropous, it takes a 180-degree turn. It is most common. In hemitropous, the orthotropous takes 90-degree turn, exactly at 90 degree. Example is ranunculus or buttercup. Fourth one is compilotropous when it is either more than 90 degree or less than 90 degree. Example here is cruciferae. Fifth one is amphitropous where it forms a horseshoe, not a regular structure, not a smooth structure, it forms a horseshoe-like structure. Example here is poppy. Next is sarkinotropous. Sarkinotropous, in this, the ovule is first in orthotropous position. It turns 180 degree, forms anatropous, and again in anatropous, it takes a turn and forms orthotropous. So it is completely taking 360-degree turn. Example of this is opuntia. Do you know whenever a word ends with genesis, it means generation. So, for example, microsporogenesis, it means generation of microspore, and here it is generation of megaspore. Why we have given the name microspore to male part and megaspore to female? Because male part is only carrying the gamete and some nutrition for itself. Other than that, it is not responsible for the development of zygote. It is only traveling from male to female part. But in female, it stores, it fertilizes there, it forms zygote there, and further development is taking in the female. That's why we are saying here the female is megaspore, right? So in microsporogenesis, you know that microspore forms from sporogenous tissue, right? Here also, the formation of megaspore mother cell will take from sporogenous tissue. But before to that, let's start from the primitive structure of ovule. So in primitive structure of ovule, the hypodermal cell of primitive ovule forms or divides to form archegonial cell. Archegonial cell. This archegonial cell can either directly form megaspore mother cell or it can divide divide very cleanly to form sporogenous cell, which then divide meristematically and forms megaspore mother cell. So here you are able to see that archegonial can either directly form megaspore mother cell or it can form sporogenous cell and then to megaspore mother cell. So ultimately we have to form megaspore mother cell. Till the stage of formation of megaspore mother cell, you're not able to see any kind of meiosis here. So all of these structure are in diploid or whatever the parents are in. Generally, we are talking about angiosperm, so they are generally diploid, generally, right? This megaspore mother cells undergo meiosis and forms a linear tetrad of 4 cells. And this linear tetrad formed at micropylar end and chalazal end. One is at the topmost micropylar end and another is at the chalazal end. In microsporogenesis, you have learned 5 types of tetrad: the tetrahedral, isobilateral, decussate, T-shaped, and linear. But here in megaspore development, only one type of tetrad is formed, that is linear. One cell is towards the micropylar and one is towards the chalazal end. But unlike in microsporogenesis, where all the 4 tetrads were functional, here only 3 will be functional. Those at the those at the micropylar end will degenerate. These 3 will degenerate, and only one at chalazal end will undergo following division, undergo following growth process. Right? So let's take this cell. This is the gamete. Now this gamete is of N, haploid type, right? So this haploid type will undergo mitotic division. Mitotic division. This is your megaspore. So this will undergo mitotic division. In mitotic division, here first only nucleus is dividing. Right now, no cell wall, no cell membrane is formed. Only nucleus is dividing right now. So nucleus will divide into two nuclei stage, and both will move toward the opposite pole. Now it will divide again mitotically and form 4 nuclei stage. 4 nuclei stage. Again undergo mitotic division and form 8 nuclei stage. 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei stage. Remember, at this stage, till this stage, there is no cell membrane division. Only nucleus is dividing. Cell membrane is complete, one single for all of them. Right? At this 8 nuclei stage, the cell membrane now divides. Cell membrane now is laid, and this is your ovary. These are your 3 antipodal cell, and these are your 2 synergids, 1 egg, and 2 nuclei for central cell. This is your central cell. So this stage is 7-celled 8 nuclei. How is this possible? See, antipodal cell, 3 cell, 3 nuclei. Okay, synergids, two cell, two nuclei, each having their own single nuclei. Egg, one cell, one nuclei. But in central cell, there is one cell, two nuclei. That is n plus n. Here n, n, all are in haploid condition. So how many cell? 1, 2, 3, 4, 5, 6, 7. 7 cell, 3, 4, 5, 6, 7, 8. 8 nucleus. This stage is known as 7-celled 8 nucleus stage. Okay. But this is when there is monosporic condition, when single spore is functional and is moving for the further mitotic division. What happens when more than one spore is functional, and those conditions are known as bisporic, tetrasporic or polysporic. More than one spore is functional, then it is bisporic. When two spores are functional, the example of this is allium. And tetrasporic, when all four spores are functional. The example of this is Drasa, Drusa and Pepperonia. Okay. So now let's move to the structure of ovary. Let's see how. Now let's see what is the structure of ovary. And with this, we will complete our pre-fertilization event. So now let's take a look on the structure of ovary. Ovary is generally 7-celled, 8 nucleated stage. 7-cell, 8 nucleated stage. Now look at those 7-cell 8 nucleated stage. These are two synergids. Synergids, along with synergids, there are filiform apparatus which is present to guide the pollen tube and male gamete towards the ovary. Right? Then there is egg. These both are of N condition. N condition. Antipodal cell at the opposite end. 3 antipodal cell. All of N, all with haploid nuclei. And one central cell with 2 polar nuclei, that is N plus N. Central cell is having 2 nuclei. Right? So this was the structure of megaspore, and this was the process of megasporogenesis. What we have learned today: megaspore, for structure of ovule, different types of ovule, types of integuments, how megaspore is formed from megaspore mother cell, and what are the different divisions that it undergo before the formation of ovary, that is 1 meiosis and 3 mitotic division to undergo to form 7-celled and 8 nuclear structure. In next class, we are going to study about pollination and fertilization event. Till then, revise this and practice questions. So for more information, you can pin down the comment. Thank you.